nanohub.org.
Online simulation, and more for nanotechnology.
Happy to have Aagam Shah as our speaker. She's going to talk about image analysis and segmentation. Aagam is a grad student at the University of Illinois at Urbana-Champaign in material science. He works with Professor Sameh Tawfik and Professor Elif Ertekin, who's here with us. And his area of work is synthesis of graphene by chemical vapor deposition. And he's going to tell us about tools that he's been developing to analyze the structures. And I'm excited about this talk for a couple of reasons. A lot of people think that data analysis and these type of tools are things for modelers. And Aagam actually does experiments. And it goes to show how important these tools are for everyone doing research, not just for simulation people. And so I'm looking forward to the presentation. For all the participants, there's a Q&A little thing in WebEx, so use that, type your questions during the presentation, and we will have a little bit of time for Q&A at the end of the presentation. So without further ado, Aagam, take it away.
Thank you very much, Ale, for that introduction. So hello, everyone. Good morning, afternoon, or evening, wherever you are. Thank you for signing up. I'm Aagam Shah. I'm a grad student at the University of Illinois at Urbana-Champaign, and I'm part of this team called Graphene Rescue, which essentially supports platforms for community sharing of data related to the synthesis of graphene by chemical vapor deposition. It is part of the NSF-funded nano manufacturing node. This is going to be the first of 2 workshops where we explore various clustering methods to segment microscopy images. We will be working with images of graphene, but these techniques can be used generally for most things. So, time, let's get on with it. So what is graphene? It is a 2-dimensional sheet that are arranged in a hexagonal lattice or a honeycomb lattice, and it has some very desirable properties like high strength and good electrical conductivity, with applications ranging from flexible electronics to transparent conductors and sensors. Next, we're going to look at a few graphene microscopy images, just for those who haven't seen them before. As you can see, these hexagonal regions generally are what graphene looks like. The rest is either a contaminant or the substrate. Every now and then you have that have slightly different shapes and are not hexagonal. And as you can see, there are many different contrasts. So the challenge is to be able to come up with a technique that can deal with all these variations. So segmentation, it is a way of separating an image into regions that contain shared attributes. So in this image, we should be able to differentiate a person from a car and from a handbag. That's image segmentation. In our case, we would be separating graphene from the substrate, or everything else rather. So the goal is to analyze each pixel, determine whether that particular pixel corresponds to graphene or not graphene. So we don't really care at this point whether it's substrate or contaminant, but as long as it's not graphene, it's one segment and the other segment is graphene itself. Now, as humans, we can usually understand and qualitatively say that, all right, this is graphene. But there are 2 reasons we need segmentation, especially automated segmentation. The first is to have a quantitative idea of things like the percentage of area covered or how hexagonal these domains are, or the size of these domains. And the second is the automation side, where we as humans can only deal with a few images, but we automate this process, we should be able to deal with hundreds of images at the same time, which opens the doors to many other machine learning applications. In today's topics, we're going to go through two approaches to image segmentation. The first one is template matching, and the next one is k-means clustering. Both of these require some pre-processing of the image, and we'll start with that. So we first, in pre-processing, we first take an image and divide it up into windows. Now, just for those who are again new to Graphene, these regions here are all Graphene, and all these regions with the white dots and all of that is the substrate and or contaminants. So yeah, let's get back to pre-processing. So the first thing we do is divide this image into windows. Now each window represents a vector of pixel intensities. For simplicity, let's take each window, let's consider that each window has only 2 pixels. More realistically, each window would contain several hundred pixels, but I will show you that on the next slide. For now, let's consider them to contain only 2 pixels. Next, we write down the pixel intensities for each of the pixels and create a vector for each one of these windows. Now, once we represent them vector of pixel intensities, we plot all of these vectors on a graph. Now, since we have only 2 pixels here, the first pixel can be plotted on the x-axis, the second pixel can be plotted on the y-axis, and we can have a 2D plot of the pixel intensities. Something like now, when we classify these intensity vectors in this plot, generally tend to see that they form clusters, and the goal is going to be to identify which one of these clusters or which 2 of these clusters is graphene and which ones are not. Now, basically, these windows have n cross n pixels, so they look something like this. So how do we deal with creating a vector for these? We flatten them out. So we take these first n pixels and then the next n, then the next n, and the next n, and we create a vector that has n square values or n square dimensions. So when we plot these out, we will get an n square dimensional space of intensity vectors, and instead of the 2-dimensional space that we had in the previous slide. The other difference from this between the schematic and what we really do is the way we create these windows. So here you can see these windows seem to be independent of each other, but in reality, the way we create these windows is something like we see in this animation here. We take a few pixels, say n cross n pixels in one, and create one window. Then we shift a certain number of pixels, which in this animation is 1 pixel. So we shift the next n cross n pixels is part of the second window, and so on and so forth. Now we can control the length of this shift. So we want to shift by 1 pixel to the right and then 1 pixel down, or do we want to shift 10 pixels to the right and then 10 pixels down? This length is called the stride length, and we can control this length. We will see how we do all of that when we go to the Jupyter notebook, but this is one of the things that we can control. Let's move on to the template matching technique now. The idea is that we select an area that looks like graphene. We try to find every region on the image that looks similar to that particular area. So we call that area the template. So here we've selected the area within this red box to be graphene. So blow it up and we divide the entire thing into pixels, and we flatten them out, we create the intensity vector like we've seen in the previous slides. And now again, for simplicity case, I'm going to take a 2D case. We plot them on the intensity vector space. We plot this particular template here. After pre-processing, we already had these black dots everywhere. And now we've added this one red dot here, which is a dot that represents the template. Next, we measure the distance from the red dot to each one of the black points. And then we say that, all right, if the is with less than a certain value, that particular point is graphene. And if it is more than that value, that is not graphene. So we create something like this circle around this point of all the points that are within a certain distance from the point representing the template or the red point. So these, all the points inside this region, even this one point right here, will be counted as graphene, and everything outside this circle is going to be classified as not graphene. Now the parameters that we can change in the course during the course of all of this analysis is going to be the template position. Shouldn't really affect our result by much. We just change the position to a region that represents graphene. The next is the size of the template, so the size of the window. And the last is the threshold, or the distance itself. We will see in the Jupyter notebook how each one of these parameters affects the result that we get. So without further ado, let's switch to the Jupyter notebook. For those who would like to follow along, please open this link in your browser, https://nanohub.org/tools/image segment. You should see a page which has something like this. Please click on the launch tool button. You your NanoHub to do so. So if you haven't signed up already or haven't logged in, please do that, and then click on the launch tool button right here. That should bring you to that looks something like this. So this is the landing page, and about a minute to get onto it. For those who are already here, please click on this particular link, template matching, to open Jupyter notebook for template matching. Sure, yes, I can share the link in the chat too. For those of you who are new to what Jupyter notebooks are, well, it's a neat way to write some code in Python. Each one of these sections is called a cell. So yeah, right here. So each one of these sections is called a cell. Here you can see two different types of cells rather. One which contains only text, and one which also contains some code. So we basically have two types of cells. We can run each one of the code cells independently, but the variables get carried on as we run them. So I think that's just a basic introduction to Jupyter notebooks. I hope everybody who wants to follow along is on this particular page and on this particular Jupyter notebook. So let's get started right with it. To run each cell, you can click on this button right here. You can also go here and say run cell, or you can use the shift plus enter keys on shift and enter keys on your keyboard to run that particular cell. So first we're going to begin with the libraries. We're going to import all the relevant libraries from the cell. I'd like to mention the CV2 library. This is the computer vision library that allows us to perform cool functions like template matching. And it's actually a very, very powerful. People who are interested in image analysis can look it up and see all its functions. It's pretty amazing. Next, we're going to import the test image and display the image itself. So this is what the image looks like. As you can see, the darker regions are graphene, and the lighter regions and the white dots are all the substrate and all contaminants. We define the relevant parameters. So like I mentioned in the presentation, we have three parameters. One is the template size, or the size of the window. So we've taken that to be 20 pixels cross 20 pixels here. The next is the threshold, or the distance. Now, you will see later on during this workshop that this distance is actually a relative distance. And so we've taken that to be 0.25, which can be anything from 0 to 1. And the last one is the position of the template. As you can see, at 275 and 500, which is 275 somewhere here, and 500, which is somewhere here. So this is graphene. So we've selected this position to be the position where the template starts, or the top left corner of the template. Let's run the cell. This particular cell is not required as part of the segmentation process. It's just to view where our template is. So to just bring it together with the slides, you can see a small red box right here. Not sure if I'm zoomed in enough. Let me zoom in. I think I forgot to do that. All right, I think this is better. Sorry about that. So yeah, getting back to the presentation, yeah, this particular region to be our template, and next display the template itself and assign all these values to a particular array. Create an array and assign the pixel values to it. We do the actual template matching. So cv2.match template. So match template is the function that allows you to do it. It takes three inputs. The first is the image itself. The next is the template, and the last is a statistical technique to determine the distance between the points. There are many different ways to determine distances in an n-dimensional graph. So here we've chosen the square difference, the mean square difference distance, and it's normalized. So this is what I meant when I said that all the distances, our threshold can lie between 0 and 1. There are many other techniques that you can use. There is, I have put in the link to the match template, the documentation for the match template function here, and you can view all the different techniques on this page and change it as per your requirements. Let's run the cell, and this is something like a heat map of the distance between the and every point in the image. As you can see, so the darker regions have lesser distance, and as the pixel becomes whiter, the distance increases. So as you can see, these regions seem to have a lesser distance from the template, and that is what we'd expected. So next we apply the binary filter, which is, we say that every pixel which has distance that is less than a particular threshold is going to be considered graphene, and everything else is going to be considered as not graphene. We apply a simple binary mask, a binary filter, sorry, and we get this result. All the white regions here are graphene and all the black regions here are not graphene. So we have successfully segmented our original image and come up with uh, an image that has two separate regions, one which has graphene and one which doesn't. Just to recap what we did right now, we started with an image and selected a template, a region that has graphene, then assigned that template to a variable and use the match template function to the heat map of the distances of every single point to the template itself, and then we applied a binary filter which regions are graphene and which are not. I would like to point out at this point, physically do a lot of pre-processing since the match template function does a lot of that for us. Here the stride length is 1 everywhere, but we will do a lot more of that a lot more of the pre-processing in the k-means Jupyter notebook. Now let's vary the two parameters that we had. One is the template size or the size of the window, and the next is the threshold. And let's see how it affects our result. So first we create a function that does all of the above steps for us. It just takes in the size of our template, it takes in the threshold and the template position, and returns a mask that looks something like this. So let's run this cell. And next we vary the template size from 10 cross 10 pixels to 20 cross 20 pixels, and then eventually to 30 cross 30 pixels. We leave the threshold to be the same, 0.25 as before, and the position of the template to be the same. Let me run this again since it's so what we see here is that for a template size of 20, we have the same result as we had before, since that's what we did in the beginning. For a template size of 10, we have a very odd result. The substrate seems to be considered to be graphene, and it's an entire mess. This is because when we decrease the template size and we make it very, very small, we lose the essence of what graphene is. We zoom in too much and we're no longer able to really sample graphene well. On the other hand, when the template size becomes too large, we start losing some detail, we start having rounder edges, and we are not able to segment graphene very, very accurately. So we want to try and find the right template size for our image. The next is the threshold. We're going to vary the threshold from 0.2 to 0.25 and then eventually to 0.3. We're going to leave the template size and the position is the same. And from the result, we can see something similar to what we would expect. At 0.25, we have a result that's the same as earlier. At a lower threshold, a lot of the points that were considered graphene here are not graphene anymore. And at 0.3, a lot of the points that were not graphene here are considered to be graphene. So this is pretty much everything that I had to tell you about template matching. I think now I can go back to the presentation and we can talk about k-means clustering. All right, so just as a recap, we first divide the image into multiple windows. We flatten each one of these windows out and make pixel intensity vectors. We plot all of these vectors on a high dimensional pixel intensity plot. And now the thing that is different from in the pre-processing from template matching between template matching and k-means is that here we also control the stride length. So we can choose how many spaces we want to move to create the next, to pick the pixels for the next window. We can have that to be 1, we can have that to be 3, 10, 100, whatever we fancy. Next, so we, as you remember, we end up with this pixel intensity plot. Again, for simplicity, I'm going to take a 2-dimensional plot. And in k-means clustering, again, we start from here. So the idea is to divide this entire plot into multiple regions, and each one of those regions is going to be considered as one cluster. So we start with these pixel intensities, and then we initialize centroids. What that means is we randomly pick a certain number of centroids and place them randomly on this plot. So the number of centroids that we pick is going to be the value of k. Here I've chosen them to be 3, and I've placed them at random positions on the plot. Now we construct something called Voronoi polyhedra. A Voronoi polyhedron is basically a polyhedron that comprises of all the points that are closest to that particular centroid. So just to give you an idea, the blue Voronoi polyhedron is something like this. All the points that lie in this blue region are closer to the blue centroid than the other 2 centroids. Same goes with the red. All the points that lie within the red region are closer to the red centroid than any of the others. And same with the green. Next, we average the points in each region. So in the blue region, we have all of these points. We average the position of all of these points. We come up with a point somewhere here. Next, we take the red region. We average all of the points here, and we find something just above this cluster. And then we average all the points in the green region. We find a point that's somewhere here. And we assign the new centroids to be those points for each one of those regions. We use these new centroid positions to recalculate the Voronoi polyhedra and get something like this. As you can see now, there are more points from this cluster within the green region, and slowly we start moving in this direction. We recalculate the mean positions and then shift the centroids and then recalculate the Voronoi polyhedra. This gives us a plot that will eventually look something like this. We have, at this point, if we average the positions of the points in each polyhedron, the position of the centroid is not going to change. This is the point where the algorithm stops, and we can now assign each one of these regions to being either graphene or not graphene. We have successfully segmented the image at this point. The parameters that we can change here is k, which is the number of clusters. We can change how many centroids we have, so we can change how many regions we want to divide the image into. The next is the tile size or the window size, and the last is the stride length. We will see how each one of these changes our result in the Jupyter notebook again. Just as a reminder, the stride length is the length that is moved when we're creating those windows before flattening them out and creating this particular pixel intensity plot. The advantages of k-means is that it's very, very fast, it's very memory efficient. We don't really need a lot of computational power to use it. And we don't have to select a template or threshold. It does everything, almost everything on its own. It's going to divide the image into a certain k number of clusters that we give it. And the drawbacks are that we need to actually specify the number of clusters. It's not entirely automated. And we also need to assign the labels. So it will not tell us which region is graphene or not graphene. It will just say that this set of points is similar to each other, and this other set of points is also similar to each other. But it's not going to tell you what's graphene and what's not. And the last issue is that when you have images which have things like concave shaped blobs, k-means clustering fails, because no matter how much you try, you're not going to be able to come up with any way to divide this image into the actual clusters that are present. I think we can, let's move back to the Jupyter notebook again. This time, again, for those who've closed their tabs, please open nanohub.org/tools/segment. And let's particular page right here. Please click on the k-means clustering link here, and that should open a Jupyter notebook like this. I'm gonna give everybody a few seconds to get to this page. Again, for those who want to follow me, you can click on the run button right here, or the one right here, to run every cell that has code in it. You can also press Shift Enter on your keyboard to run the cell. So we begin again by all the relevant libraries. The important ones that I'd like to mention here are the k-means and the mini-batch k-means libraries. These are the libraries that allow us to perform the k-means clustering. I will explain what mini-batch k-means is as we come to it. Again, we start by importing the image. We print the resolution of the image and we display the image itself. It's just for our convenience. There's no significance to it per se. This is what the image looks like. You can see there's a lot of graphene on this image. These light gray regions are the substrate. Graphene also has different contrasts all throughout this image, and it's not an easy image to segment. So let's try and segment this image. We now define all our parameters. So the first is the window size. Here we take the window size to be 30 pixels plus 30 pixels. Next is the number of clusters. We take that to be 5. This is the value of k. Stride length is something that we hadn't done in the template matching. So the stride length is going to be 3 pixels this time. So we're going to shift 3 pixels between windows. And the last one is the seed. Now the seed value is not something that you necessarily need. It can be any random integer. If we don't specify one, the function will just pick any random value. But this value is used to randomly place the centroids at a particular position. So if we change this value, the position of the centroids is going to change. But generally our final result won't change by much. So let's cell. Next, we pre-process the image. So the first thing we do is pad the image. We define a function that adds a few dummy pixels to the image. Why do we need to do this? This is primarily because we need an integer number of tiles. So let's take an example where we have an image that is 8 pixels cross 8 pixels. And we have a window size of 5 pixels cross 5 pixels and a stride length of 2. So we take pixel 1 to pixel 5 in the first window, then we move 2 pixels over. So we take pixel 3 to pixel 7 in the next window. Then we move 2 pixels further. We take pixel 5 to pixel 9, but there is no pixel 9. So what we do is we add that ninth column and row of pixels, and we just assign dummy values to them, and we create the integer number of windows that we need. So this is that does that for us. In the next cell, we're going to define this variable called x. What this variable is it's an array of each one of these windows. So our window is 30 cross 30. So it's going to take the first window, assign it to the first row and first column. Then it'll move to the right, which is 3 in this case. So move 3 pixels to the right, take the 30 cross 30 pixels, and assign that to the second value, which is the second column and in the first row, and so on and so forth. It's going to keep moving to the end of the image on the right, then move 3 pixels down, and it's going to cover the entire image. So X is basically an array that contains these windows. Next, we have the mass dimension. This is basically the number of this mass dimension contains the value of the number of windows that we have. So it's going to contain 2 values. One is the number of windows in the x direction and the number in the y direction. The last thing that we're going to do is flatten out each one of these windows. So we use the reshape function for that, and that's pretty much what every single line in the cell does. Next, we're going to run the model itself. So we first define the k-means model. We give it the number of clusters, and we give it the initial seed value that we had defined earlier, which is just a random integer. If we do not give it this value, it will pick an integer at random. Next, we fit all of this. We fit this particular model to our data, which we've made here, and the percent time that I've added at the beginning here is going to display the time that it takes to run this particular line of code. Next, we're going to do the same for mini batch k-means. So I'll take this moment to explain what mini batch k-means is. If you recall from the presentation, in k-means clustering, we take an average of all the points that lie within a particular Voronoi polyhedron. Mini-batch k-means we randomly pick a few of those, not all of them. We randomly pick some of them and average only over those. And while we iterate, we don't always calculate over every single point. Although this sounds like it will be erroneous, we will soon see that our result is not very different from what we get from the original k-means clustering, but the computational time and the resources taken is significantly lesser. So let's wait for the k-means cell to run. I think we can also run the mini batch k-means for now. just so that they're sequentially placed. As you can see, the k-means is taking very long. Although we've started running the mini batch k-means, it's gonna wait till it finishes the previous cell. That's the way Jupyter notebooks work.
Aagam, you need to have a joke for this kind of occasion. Tell a story. One of the interesting things, let me chime in. We have over 100 participants, and a lot of them are running the same simulation. So that tends to put a little bit of load on NanoHub computational resources with lots of people running at the same time. So that's why it sometimes takes a little bit longer.
That's right, because normally when I ran this before in all our trial runs, the k-means took about a minute and the mini batch k-means took about 3 seconds. So we can see the difference right here when everybody's using this off NanoHub. But still, to reiterate my point, k-means takes about 3 minutes this time, and mini batch k-means takes about 11 seconds. So we can see the difference in the computational time and power between the two. We will soon see what the difference is in the result. So next, once we fit all of our, we fit everything to the k-means and the mini batch k-means model, we post-process the data. So what we get after this is a bunch of attributes that are assigned to variables here. So the k-means variable here and the mini batch k-means variable that we created here. The relevant one right now is the variable called labels. This is going to be a vector that contains all of the values. So it's going to contain values from 0 to 4, so 0, 1, 2, 3, 4. These are 5 different values, each value representing one cluster. And it's going to say that this particular pixel is this cluster, or rather window. This particular window is this cluster. This particular window is, say, cluster number 3. The next one is cluster number 5, something like that. So we're going to run this and do the same thing for mini batch k-means. What we do in the first line is reshape it to get our x cross y again. This is originally just a one-dimensional vector, and then we create an image object from that array, the two-dimensional array that we've created on the first line here. We do the same. We do that for both the k-means and the mini batch k-means, and we eventually assign them to clusters and resize the image to the size of the original image that we had, or the image that we started with. This line right here is not required for the segmentation process per se. This is just so that when we plot the final results, we can see the colors of the clusters to be the same between k-means and mini batch k-means. So let's plot our segmented images. So as you can see here, this image has been segmented. Just FYI, the white regions in both the images and these light gray regions right here at the edge of all the white regions are graphene, and everything else is, well, and also the black region. Yes, the white, the black, and the light gray. these three regions are graphene and everything else is not graphene. If you look at these images at the first glance, there is practically no difference between the k-means and the mini-batch k-means. But if you really pay some attention, you will see that this region right here and this region right here is slightly different. The region right here and the one right here is slightly different. So you would see some very, very small differences between k-means and mini-batch k-means. But the difference in computational time is very, very large. So generally in most applications, we try to use mini-batch k-means because it saves us a lot of time and computational power. In the next one, this is just for visual aid. I'm going to plot each one of these clusters in a separate image. You can see the different regions in different images. Now we're going to vary the parameters. So just to recap everything that we've done so far, we started the image and defined all our parameters. So we defined the window size, the number of clusters, the stride, and also the seed in this case. We then give a function that adds the image and adds a few dummy pixels to the sides so that we get integer number of windows. We pre-processed our image. We picked those windows and we flattened them out, created those vectors. We passed to our k-means model, fit all of that, fit all of our data. Then we took the attributes from these k-means and the mini-batch k-means variables. We reshaped them to form our mass dimensions, converted them into image objects, and resized them to the image that we had in the beginning. Plotted our results. So when we start varying the parameters, we're going to create a function. Now we're going to use mini-batch k-means just because it's much faster. At this point, those who are following me, I would ask you to run all of the cells from this point forth together. So you can go to cell and run all below. While you're running, please scroll back up to here. So yeah, we first defined a function that does everything that we did above for us in one go and returns an array of the clusters that we can plot. And we start varying the number of clusters. We first try 2 clusters, then 5, and then 8. We leave the window size to be 30 pixels cross 30 pixels, and the stride length to be 3 pixels still. And we keep the seed as the same just so that we don't really have any variation. I've also added the time function here so that we can see any change in the computation time. The first one is the number of clusters when we take them as 2. Since this image has a decent contrast between graphene and the substrate, we can see that when we have only 2 clusters, the segmentation is done pretty well, and for the most part, we know what is graphene and what is not graphene. The edges are a little rough, but we can tune all of that. When we take 5 clusters, I feel there are too many clusters, and we don't really need so many, but we can still identify which clusters are graphene and which are not. When we increase the clusters to 8, we have clusters that are that we cannot really relate to this image, and it's just too much information or too much segregation, and we don't really need all of that. So generally, the idea is that we try to keep the number of clusters close to the number of segments that we want to create. In this case, we have 2 segments. One is graphene and the other is not graphene. So we try to keep it to 2 clusters or maybe 3 clusters, but not all the way up to 8. As you can see, the computation time doesn't change much because the number of clusters doesn't really change the math that we have to do. It's still going to be the same number of dimensions because the window size is the same, and it's still going to be, we're still going to skip the same number of pixels because the stride length is the same. So the number of averages and the number of calculations and all that is almost the same. There isn't any significant change in the computation time. To vary the stride length, we're going to start with the stride length of 3 pixels, then 10 pixels, and then 25 pixels. Here we've taken the number of clusters to be 3 so that we no longer have to see something like this and kind of scary. So let's do this. As you see, when we increase the stride length, our image starts pixelating and we start seeing 1990s kind of image. So this is primarily because the larger number of pixels we skip, the more the rougher our result is going to be. We won't really see as much detail as we will with a smaller stride length. But you also see that increasing stride length, we have a significantly decreasing computational time. So we need to find the right balance. We don't really want a very high computational time. So if our stride length is 1, our computation time is going to shoot up. But if our stride length is 10, we will get a pixelated image, but with a very good computation time. So we're going to try to find the right balance again here. Again, the computation time decreases because we have lesser number of windows that we create. If we increase the stride length, the number of windows will decrease, which is going to decrease the total number of points that we have on our intensity space plot, and that's going to decrease the number of calculations that we need to make. The final one is the window size. We're going to vary the window size from 10 pixels cross 10 pixels to 30 pixels cross 30 pixels, and eventually 50 pixels cross 50 pixels. When we have a smaller window size, you can see that there is a lot more detail. But because the window size is too small, similar to what we had in template matching, we lose a sense of what graphene really is and have some very weird regions that show up. Luckily, in this case, we still have decent clusters that can say what is graphene and what is not. But if the window size is too small, we will have too much detail, or we will lose the sense of what graphene is. Here we have a decent window size. All the regions that are white are not graphene, and the gray and the black regions are graphene, which is a reasonable window size, I feel. And the last one is where the window size is 50 cross 50. Here we lose a lot of the detail that comes with the 30 cross 30. We have rounder edges, and we have a slightly more erroneous fit. When you look at the computation times, we see that when the window size is 10, our computation time is very less and the time increases very rapidly. When the window size is 50 pixels, we have a computation time of almost 4 minutes. This is because as our window size increases, the number of dimensions in our pixel intensity plot increase in a square manner, so in the n square manner. And this is going to significantly increase our computation time. So we try to stick to a decent number of decent window size where it's not too small to either have too much detail or lose the essence of graphene, but it's not too large that we have rounded images and computation time. With this, I'll let you end the Jupyter notebook on k-means clustering. Let's go back to the presentation for a quick show. So this is primarily for those who don't really want to go through all of this code. We have a tool on NanoHub called the SEM image processing tool, which does everything that we have done in this workshop as a software. So you can take a template, place it here and select the threshold and the template size and come up with a mask or a segmented image. So you can go to this particular link, nanohub.org/tools/GSA image, or you can look for SEM image processing tool on NanoHub. I would like to thank everyone for attending this workshop. We have another workshop next week on supervised clustering methods to segment graphene, to segment microscopy images of graphene. We will be learning how to make deep neural networks and use that to segment our images. So we just go back to this landing page. For now, these two links do not work, but next week you will be able to click on these links, and that's where we will have our Jupyter notebooks for the workshop. Thank you for attending this workshop, and thank you for your time again. And I think I'm open to questions now.
Thanks, Aagam, for a very nice presentation.